When you think of luxury food, what kinds of food do you think about? You may think about steak or even caviar, but today I'm going to talk to you about a species used in many luxurious dishes, the lobster. Lobster is a highly sought and highly fished species of crustacean that is often fished for in great numbers by many fisheries. The Caribbean spiny lobster, or Panulirus argus, is one species of lobster that is a high priority target of said fisheries. This species of lobster is fished along the rims of the western Atlantic Ocean, all the way from the north of Brazil in the southern hemisphere to the Gulf of Mexico in the northern hemisphere. The Caribbean spiny lobster is particularly abundant in the Caribbean Sea, and it is heavily harvested in the Florida Keys in the United States. So how can the same species of lobster be found all across this long stretch of the Atlantic Ocean? Perhaps you're thinking that the lobster walks there on the sea floor over a long period of time, but actually, evidence shows that this wide geographical distribution has to do with the lobster's life cycle. After mating in coral reefs, Panulirus argus females brood embryos that develop and hatch as larvae. Panulirus argus larvae flow in the water column for long periods of time and go through 10 consecutive planktonic stages. The larval development of this species can last anywhere between 4 months and 18 months, giving the lobster a lot of time to freely float around the open sea. In this time, it's likely that the larvae will drift in the water, often very far from where they were originally hatched. Because of this, different populations in completely different locations can still be connected. In order to understand this connectivity among distant populations, we have to generate genomic resources. One such resource is the mitochondrial genome that provides markers with which to understand interpopulation genetic connectivity. Recently, the mitochondrial genome of Panulirus argus was assembled using next-generation sequencing technologies in the laboratory of Dr. Antonio Biza here at Clemson University. In this video, I will describe the mitochondrial genome of the Caribbean spiny lobster, Panulirus argus. The mitochondrial genome for Panulirus argus is 15,739 base pairs in length and is comprised of 13 protein-coding genes, 2 ribosomal RNA genes, and 22 transfer RNA genes. When describing gene location, we can use the terms L-strand, meaning light strand, and H-strand, meaning heavy strand, in order to describe which strand of DNA a gene is contained in. In short, the heavy strand is the strand that contains more genes in total, and the light strand is the strand that contains less. With this in mind, I'd like to point out that only 4 protein-coding genes, 8 transfer RNA genes, and the 2 ribosomal RNA genes are encoded on the L strand, while the remaining 9 protein-coding genes and 14 transfer RNA genes are encoded on the H strand. The mitochondrial genome of this species is quite compact, meaning that there are not many intergenic spaces or overlap between genes. For instance, there are a total of only 11 gene junctions that exhibit intergenic spaces, and each of these spaces range from only 1 base pair up to 32 base pairs. One space that I'd like to point out is a single long intergenic space. This space is 801 base pairs in length and is assumed to be the D loop, or the control region. The control region is involved in mitochondrial genome replication in animals. However, we know little structural and functional details about this region in crustaceans. Now let's talk about the protein-coding genes mentioned earlier. As a reminder, there are 13 of these found to be encoded in the mitochondrial genome. We find that 12 out of these 13 genes have typical start codons, meaning that they start with ATG or ATC, while one gene, COX1, utilized the start codon CGA. This atypical start codon has been previously noted in other decapod crustaceans. As for stop codons, only 9 of these genes ended with complete and conventional stop codons. 6 of them ended with TAA, 2 ended with TAG, and 1 ended with TGA. These 4 genes actually terminated with the stop codon T. This incomplete stop codon is often observed in other arthropod organisms. This table describes the many different codon frequencies observed in the mitochondrial genome of Panulirus argus. Feel free to pause the video if you would like to study it further. The only thing that I would like to mention is that this mitochondrial genome contained an adenine-thymine bias. The base composition is as follows: adenine with 32.9%, thymine at 29.7%, cytosine at 22.7%, and guanine at 14.6%. It is worth noting that this adenine and thymine bias has been previously reported in other crustaceans, including spiny lobsters. Let's now talk a little bit about selection pressure in protein-coding genes. A parameter called the Ka/Ks ratio is often used to estimate whether mutations in a genome are neutral, beneficial, or purifying under selection. Looking at Figure 2, you can see that the Ka/Ks ratio for all of the protein-coding genes is less than 1. This indicates that the genes are evolving under purifying selection. In fact, the Ka/Ks ratio for the genes CYTB and COX1 were an entire order of magnitude lower than the other genes. This may indicate that these two genes have heavy evolutionary constraints that the remaining genes may not exhibit. Let's now look at the structure of the transfer RNA genes that was found. Firstly, the transfer RNA genes that were analyzed all ranged from 64 base pairs to 71 base pairs in length. And by looking at this figure, you can see that almost all 22 of them exhibit a cloverleaf secondary structure. This cloverleaf structure is the typical shape of transfer RNA genes in other crustaceans. All of the transfer RNA genes formed a cloverleaf structure, except for phenylalanine, which is missing one arm. I would like to now talk about the D-loop, which was mentioned earlier on. This is an 801 base pair long intergenic space in the mitochondrial genome. As you can see, it is located between the 12S ribosomal RNA gene and the tRNA isoleucine transfer RNA gene. As with most of the genome, this section was adenine and thymine rich. On contrary to some other crustaceans, this D-loop did not exhibit tandemly repeating sequences. Instead, two adenine-thymine rich motifs were detected, one in the H strand and one in the L strand. Finally, we can use the protein-coding genes to explore phylogenetic relationships between decapod crustaceans. The phylogenetic tree places Panulirus argus in a monophyletic clade with another species, Panulirus japonicus. Other well-supported decapod clades include Brachyura, Onamura, Gebedea, Glyphidia, Ostacidia, Caridia, and Panioidea. Oxidia was moderately supported, and support diminished towards the root of the phylogenetic tree. This analysis suggests that protein-coding genes in mitochondrial genomes are useful for studying phylogenetic relationships among distantly related groups of decapod crustaceans. In summary, I have described the genomic features of the recently assembled mitochondrial genome of the Caribbean spiny lobster Panulirus argus. This species is a heavily fished and overexploited species of lobster in the Caribbean region. The assembly and study of this mitochondrial genome will improve our understanding of the metapopulation connectivity in this remarkable and tasty species.